Hello, and welcome to this VJHemOnc podcast, where we will be sharing a roundtable discussion from the 8th International Workshop on CAR-T and bispecifics. In this episode, experts discuss advances in lymphoma cellular therapy, including NK cell therapies, allogeneic and in vivo CAR-T cell approaches, and strategies to improve safety, manufacturing, and access. We will then turn to the evolving role of T-cell engagers in follicular lymphoma and large B-cell lymphoma, highlighting their potential use in earlier lines of therapy and the challenges of integrating these approaches into clinical practice. This podcast is part of the CAR-T and cellular therapy focus channel on VJHemOnc, which is supported by Legend Biotech. This supporter has no influence over production of the content. This podcast is intended for healthcare professionals only. So, without further ado, let's begin.
Hello, I'm Krish Patel from the Sarah Cannon Research Institute in Nashville. We just finished a great session on CAR T cells, cellular therapy in general, in lymphomas, and I'm joined by my esteemed colleagues who will introduce themselves.
Hello, everybody. My name is Paolo Strati. I'm a lymphoma investigator at the University of Texas in MD Anderson Cancer Center, Houston, Texas.
And I'm Michael Jain. I'm a lymphoma doctor at Moffitt Cancer Center here in Tampa, Florida.
So we just finished this session really focused on identifying some of the trends in cellular therapies and lymphomas. So we had a great kind of forum from what might be coming next in terms of technology to Mike, you talked about modulating toxicity. So maybe Paolo, I'll turn it to you first. We talked about natural killer cells as a platform for immune effector cell therapy. Can you tell us a little bit about what we've learned so far and where you think the field can take this?
Yeah, it was very exciting to be able to talk about the use of NK cell as a cellular therapy source for patients with B-cell lymphoma. As a reminder, currently we utilize autologous T cells for patients with relapsed refractory B-cell indolent and aggressive lymphoma. So NK cell is really still an experimental source, and we can either utilize cord blood-derived NK cells, or what I presented today was mainly induced pluripotent stem cells that have the advantage over cord blood-derived NK cells to generate billions of cells in a short amount of time and with very homogeneous biology. I focus mainly on two clinical trials because those are the two that have mature enough clinical data for public sharing, FT516 and FT596. And the take-home message is that, so in the first case, we had iPSC-derived NK cells that was engineered to express a high affinity and non-cleavable CD16 for better anti-tumoral activity. And it was very safe. We really saw almost no cytokine release syndrome, definitely no neurotoxicity, and no GVHD, despite this being an allogeneic product. With FT596, we saw similar data. And as a reminder, the main difference between 516 and 596 was the addition of a CAR, target Z19, but also an interleukin-15 receptor fusion protein so that the NK cells could be more persistent. And there was significant activity also in patients with aggressive B-cell lymphoma. So in both trials, patients were heavily pre-treated with a median number of prior systemic lines of treatment of 4 or above. And despite that, even patients with aggressive B-cell lymphoma were able to achieve durable remissions in about 30% of cases. So definitely we do have a product. In this case, again, non-T-cell based, so not coming with all the typical toxicities that we see with T-cells, such as CRS or ICANS, and most importantly, allogeneic, that faces a very important dilemma that we currently have in the field, as these patients with aggressive B-cell lymphoma, on one end, sometimes they can't wait for the manufacturing of an autologous CAR, but also if they're in the community, they may just struggle to adjust to the logistics the autologous CAR require. And these products seem to be safe and effective, but we need to work more on the persistence. So to the second part of your question, the future currently is trying to further engineer these iPSC-derived NK cells to make them more persistent. For example, we have created a new product called 522, but we had knocked out CD38 to try to minimize the fratricide that currently NK cells experience with each other. But also there was an addition of an ADR, a receptor recognizing allo defense to 4-1BB, and so minimizing allo reactivity and potentially even minimizing the dependency on lymphodepleting chemo. That is probably the next step that we need to do in cellular therapy, try to get away with cellular treatments that don't require any chemotherapy at all.
Yeah, no, you really summarized that nicely, Paolo. I think one of the things I'm struck by in the NK cell platform is how safe the therapies were. Very, very little, if no CRS, no ICANS. Kind of juxtaposing that with what you presented, Mike, We know autologous CAR T cells are a great value, right? Curative treatment in lymphoma, but they can be beleaguered by toxicity. Tell us a little bit about the work that you've been doing at Moffitt to try to help modulate that toxicity using medicines that we, already have access to. I thought that was fascinating.
Yeah, so we've been able to treat standard of care patients with CAR T-cell therapy almost a decade now. Mm-hmm. And it's quite different, when you go from mouse models to humans. We're all physicians and our patients don't look like healthy mice, right? They can be very sick, inflamed. Inflammation is a big part of the cancer syndrome. Patients can lose weight, and get blood clots. And so it turns out that this is actually a pretty important predictor for whether or not when you put an engineered immune cell into a patient, that baseline inflammation tends to flare up after the CAR T-cell therapy. And so some patients have more inflammation than others. And is there a way that we can use known anti-inflammatory agents to try and bring down that inflammation and thus, really get good outcomes in all patients? And so one class of drugs that's been used a lot, particularly in the transplant field, but also all around immunity, are the JAK inhibitors. And so the study that we did was to take patients who had a lot of baseline inflammation, treat them with JAK inhibitors before and during CAR T-cell therapy. Mm-hmm. And what we found is that it indeed reduced the toxicities, but what we were really going for is to try and have patients relapse less. Because we think the same inflammation that causes the toxicities are also suppressing the T cells, the CAR T cells that are doing the anti-tumor aspects. And so then we did some sort of very detailed studies using something called single-cell RNA sequencing to look at, okay, how are these anti-inflammatory molecules, the JAK inhibitors, affecting various parts of a patient's immunity and inflammation? And so I think this is very interesting and potential future avenue for combining known drugs with our existing therapies, to do better than we have over the last 9 years.
Yeah, no, I think that was really fascinating. Repurposing a drug to both potentially improve efficacy, maybe also modulate safety. Also in our session, we talked a little bit about allo CARs, and then the session after ours, we talked about in vivo CAR T cells. And so we recognize not every patient can get to an autologous CAR T cell. Access can be a challenge. Maybe I'd ask you both to share, what do you think about allo CARs and in vivo CARs? They seem, again, very, very exciting platforms. What are some of the take-homes that we can take away from those sessions?
Yeah, maybe I'll go first. Definitely both allogeneic CAR or in vivo CAR, both address highly unmet need, both from a biological and clinical perspective. So biologically speaking, one major limitation of any autologous product is that we are collecting immune cells for patients who are previously treated. Mm-hmm. And as such, those particular T cells can be hampered by the prior systemic chemos in particular. So utilizing, of course, allogenic products or in vivo CAR, that's resolved. One additional advantage of the in vivo CAR, going back to what I was mentioning before, is that don't require lymphodepleting chemo. Mm-hmm. That instead is a major problem currently for allogenic products as we're trying to minimize alloreactivity. But I would say we all were extremely positively impacted and surprised by the very good results that we heard at ASH in multiple myeloma. And we hope that we'll see some similar results in lymphoma.
Yeah. What are your thoughts, Mike, on the in vivo platform? Is that something you're working with, and where do you think it's going to land?
Yeah, so it already felt, extremely highly technologically sophisticated to take someone's T cells, put them in a dish, genetically engineer them into CAR T cells, and then have them cure people's cancers. That already seemed amazing. But, as you pointed out, this is not perfect, and there are quite a few challenges with that therapy. Now, the next level idea being that you can just genetically engineer it directly in the patient's body seems even more futuristic. Mm-hmm. And it does seem that this is possible. You can put the virus directly into the patient or a particle and have your own T cells become genetically engineered through these gene engineering strategies. Now, this theoretically would overcome several of the problems of current CAR T cell therapy. It remains to be seen. Only small numbers of patients have been treated, but lots of companies are ramping up to do this. We have a trial at Moffitt of in vivo CAR T cells. And so we're all very, very excited about the possibilities, but it has to be proven, I think, to see whether it'll become part of our future.
Yeah, a lot of points you raised, I think both may help bring down costs, may improve accessibility, but ultimately we have to make sure that they work as effectively and are as safe as the products we have today. So thank you both for taking us through that session.
Next, we move to another rapidly evolving area of lymphoma care, T-cell engagers in follicular lymphoma and large B-cell lymphoma, and how these therapies may reshape frontline treatment strategies in the years ahead.
Hi, I'm Loretta Nastoupil, and it was a great honor today to co-chair the session on T-cell engagers and lymphoma. And very happy to have my colleagues here today. Lorenzo?
Hi, Lorenzo Falchi. I'm a lymphoma specialist at the Sloan Kettering Cancer Center in New York State.
And Jason?
Hi, Jason Westin from MD Anderson Cancer Center.
So, Lorenzo, I'm going to start with you. So, you kicked off our session talking about frontline approach in follicular lymphoma, particularly focusing on mosunetuzumab, which is CD3, CD20 bispecific antibody. So, where are we currently with the treatment landscape, and how do these studies that you sort of highlighted potentially going to change that?
Yeah, we are at a very exciting time. We're at a moment where when I sit, in the room across from my patients, I can tell them that we're on the brink of a new era where we have transformative drugs that are truly chemotherapy-free, that can have the same potential as chemotherapy, potentially. And the data that I presented today, I think, really underscore the role of mosunetuzumab as a bispecific antibody targeting CD3 and CD20 in follicular lymphoma. We at MSK used it in an investigator-initiated trial as a single agent subcutaneously, and we enrolled about 80 patients. And really, we saw response rates and duration of benefit at the moment that compare at least in equivalence, if not sometimes favorably, with chemoimmunotherapy or other standard of care approaches. Naturally, there's a difference between proof of concept and then evolutionarily speaking, like regulatory pathway or development of an agent. And so I think that what the landscape has moved towards, or the field has moved towards, is combination therapies. And bispecific antibodies, being immunoglobulin-like molecules, really lend themselves to a lot of combinatorial strategies, from chemotherapy to targeted agents to immunomodulatory agents. And so mosunetuzumab, for instance, has been coupled with BTK inhibitors such as zanubrutinib in our experience, but also lenalidomide in the sponsored trial that then engendered the currently ongoing phase 3 randomized trial of mosun-len versus standard of care chemotherapy. And that is only the first of a series of randomized controlled trials that are testing chemo-free immunotherapy-based approaches or bispecific-based approaches to standard of care chemoimmunotherapy. There are 4 of those trials, very large, designed to establish superiority. So if one or more of those trials is positive, that may very well shift the landscape.
Jason, you talked about where we've come just in the last few years in frontline large cell lymphoma. And then your task was really to say, are we going to have bispecifics in frontline large cell lymphoma? So walk us through the complexities there.
Yeah, thanks. So it's a great time for drug development and for our patients, where we've got a lot of new agents coming into heme malignancies and specifically into frontline large B-cell lymphoma. But that problem, that wonderful opportunity creates a lot of problems. And that includes having too many drugs without a clear way to select which patient should get which treatment. I think that'll be a challenge for bispecific antibodies, because we're seeing glofitamab, epcoritamab, and odronextamab all have randomized phase 3 trials in the frontline setting. I think all of them have a great chance of being positive studies. But on the other side, we won't have a clear way to choose which patient should get those treatment options versus other treatment options, which are also being evaluated in phase 3 trials. And they're also partnering a really exciting new treatment, the T-cell engagers, with 1970s chemotherapy, which theoretically could be T-cell suppressive in a way that's actually pulling the brakes a little bit on what an optimal partner could be. So it's a great time, but we really need to start thinking for the future about what are we going to do if we have all these options in the clinic? How do we select which patient benefits most?
And I think both of you highlighted the efficacy of these agents really quite compelling. How do we mitigate some of the toxicity? And so, Lorenzo, I'll start with you. In frontline follicular lymphoma, it's always a balancing act of maintaining good quality of life, because these patients are anticipated to live for decades. Is a single agent going to get us what we need? Or if we need the combination, as you suggest, to enhance that efficacy, what are the practical strategies of making sure we keep these patients safe?
Yeah, no, that's a great question, because bispecific antibodies are drugs that have a very reproducible safety profile, but where things like cytokine release syndrome is a very prevalent adverse event. And early on, this was a scary adverse event. And the early data, the phase 1 data, particularly with glofitamab or even epcoritamab, especially when this drug were not step-up dose, there was perhaps less emphasis on prophylaxis. These CRS were really severe, meaning grade 3 and higher. In follicular lymphoma, particularly with the advent of step-up dosing, subcutaneous administration, we're now at the point where, yes, CRS is seen in about half of the patients, but it's really only grade 1. And I always remind my patients and some of my colleagues that grade 1 CRS means fever, period. Grade 1 infusion-related reaction from rituximab could not manage at home. You need a nurse to manage it. Grade 1 CRS, you can manage it at home, and none of our patients comes to the clinic. This is a key ingredient if you want to disseminate a treatment in the community where with subcutaneous administration, the chair time is very limited. CRS is, I would say, controlled, and it's a cycle 1 phenomenon. It really doesn't happen beyond that, maybe cycle 2. And then after that, really patients are on a treatment that they barely feel. The emerging adverse event related to bispecific antibodies in follicular lymphoma and beyond is related to immunosuppression. And these drugs are definitely B-cell suppressive in a way that rituximab is not. But they're also probably T-cell suppressive. We just don't pay enough attention to it because we don't have an immediate assay spendable in the clinic that tells you, oh, this patient is T-cell suppressed, unless you do very advanced T-cell subset, analysis. But we know that's true because these patients get opportunistic infections and whatnot. So definitely prophylaxis with, antivirals, antibacterials, sometimes immunoglobulins, depending on the level of hypogammaglobulinemia. These agents can be associated with neutropenia, so G-CSF prophylaxis or secondary prophylaxis, certainly something to also think about, but nothing that us hematologists haven't really seen before. So I think that safety itself should not be scaring us more than chemotherapy or rituximab have in the past.
Jason, you hit on some really key aspects of we're going to have probably several trials read out in the next few years. We saw Polarix had a modest impact on PFS, no difference in overall survival, but we are starting to see that landscape evolve because the toxicity is very similar to vincristine when you replace it with polatuzumab. If we have, 4 positive studies, including tafasitamab, the naked CD19 antibody, what tools or what expertise can you share on the average treating community oncologist to help them pick a frontline therapy for large cell lymphoma?
I wish I had a short the answer to that because that's the real challenge that's going to happen for people who don't treat large cell lymphoma every day. How do you have a shorthand rule of this condition means I should use this approach? This condition means this approach. And we've tried to pull things apart looking at the cell of origin with the idea that polatuzumab might be more effective in that situation. With becoming bispecific antibodies, as of yet, we don't really have a biomarker to say this patient will or will not benefit. And so I think it's going to become increasingly complicated when we have these studies that enroll all comers, really don't have a biomarker, but they use IPI as a selector. And so I think we'll be stuck with that for the time being of those patients with the highest risk, very high LDH, very aggressive disease phenotype, will be treated with these novel approaches. But for those patients that are kind of in the middle, IPI2, IPI3, not quite as sick, it's going to be difficult for treating physicians to say, is this somebody I can just give R-CHOP to? Or is that inadequate? Do I need to use one of these targeted therapies? And if I do, which one? I think what we'll see at the ASCO meeting this summer, 2026, is that tafasitamab plus lenalidomide is going to enter the conversation as another positive frontline study when combined with R-CHOP. And that uses lenalidomide, which we know also works a little bit better in the non-GCB subtype. So even the cell of origin predictor we've used over the past few years might be more complicated. So this is a big need for our field to help treating physicians to better understand this means I should use treatment A, this means I should use treatment C. And as of right now, we don't have answers to that.
I really appreciate being able to ask both of you sort of your insights into this novel but emerging landscape. And I think this is just good news for patients.
Absolutely.
Thank you for listening. To explore more expert interviews and roundtable discussions, head over to VJHemOnc.com. Be sure to follow us on X at VJHemOnc to join in the conversation, and subscribe to the VJHemOnc podcast on Spotify, Apple Podcasts, or Podbean.